Here we're talking about modern adaptations, and I want to begin by addressing the idea that we tend to find in popular culture we've stopped evolving, right? Culture is playing a role in slowing down our evolution. From an anthropological perspective, and we have drawn on the idea of bioculturalism, we know that there's a constant interplay between biology and culture. And really, to put it bluntly, culture does not have the ability to stop human evolution. So we'll briefly talk about population genetics and what the increases in size demographically among the human species means for our evolution. So let's take a look at a few recent adaptations. One of the adaptations that we can test, for example, with even common foods around the home, we used a prop PTC test in class. This helps us identify whether or not we are a super taster or a non-taster. Now, the TAS2R gene group, this refers to a number of genes that we find on chromosome 5, for example, chromosome 7, and chromosome 12. And this particular gene that helps bind our receptors to bitter or toxic substances evolved some 200 million years ago. The function of this change genetically was to provide our receptors with allow them to bind to toxic chemicals, and that helped us identify what types of foods were in fact poisonous. So tasting prop PTC toxin, it would have permitted our ancestors, really ancient ancestors here 200 million years ago, the very small group of mammals that had been emerging, to avoid toxic food. Some other animals, they have other adaptations. For example, snakes, from what I was reading a while ago, they have the ability to sense whether or not something is toxic because they have a special gland. Well, we don't have that. Either way, tasting PTC toxin, it would have permitted us to stay away from foods that tend to be more dangerous, and that bitter substance, it's a protective mechanism that's emerged in those plants. So with the invention of farming, one of the theories here for the emergence of a gene that reduced our ability to taste bitter substances is the idea that bitter foods, eventually they were selected out, and this reduced the need to have that TAS2R gene group. And one of the things that goes along with genes is this idea of use it or lose it, because mutations can tend to build up in a particular area and then impact the ability of that gene, if it's not regularly being used, for example, for taste, to produce functional proteins. Or the other idea here is there was an advantage to eating bitter foods. Looking at some of our foods that we currently eat, they've changed radically since the invention of agriculture. So the cruciferous, for example, plant group includes broccoli, turnips, or zucchini, cucumbers. Traditionally, these were very, very bitter foods. And those cruciferous foods, they tend to be very high in PTC. And like I said, PTC, it's a very bitter substance. In fact, it's the most bitter substance known to humankind. And in these particular plants, they tend to contain glucosinolates, which inhibit the uptake of iodine. Now, there's a problem if we're not uptaking iodine. That tends to cause problems in our thyroid, which can lead to something called goiter. In addition to goiter, it can completely disrupt the functioning of the thyroid, leading to either an overactive or an underactive thyroid. So the TAS2R group, it allows individuals to sense more bitter taste. That's the high PTC that we find, and avoid foods that might inhibit that iodine uptake. There are a few advantages associated with this from an evolutionary perspective. So that ability to taste PTC, as pointed out, It may prevent us from ingesting foods that tend to be fairly high in this antithyroid compound, leading to, for example, goiter or the inability for the thyroid to function properly. On the other side of things, we know that these foods are packed full of nutrients. So it's kind of a balance between ultimately ingesting foods that are potentially harmful on one level, preventing that iodine uptake. But on the other side, we also know that these foods, for example, broccoli, they're packed full of nutrients, minerals, all kinds of vitamins, fiber, stuff that does help us from a nutritional perspective. Food, and again, it's hard for us to understand if we're eating some broccoli and saying, well, broccoli's not that bad. We do need to understand that food was very different in the past. And here we're referring to all the foods in their pre-domesticated form. So, for example, in class, I asked, what is it? And I asked this question at home, and my family looked at it, and they were hard pressed to figure out what type of a food this was. One of the responses was it the leg of an insect? Here we're looking at something that is approximately 19 millimeters in size, and it does represent perhaps one of the most ubiquitous foods that we find pretty much in everything from soda pop to other foods that use corn-based sugars. There's the answer. This is pre-domesticated corn. And the only way that you would be able to eat this pre-domesticated corn, very different than the corn that we have today, toss in some boiling water and munch on it with some butter, that wouldn't have been possible. The only way to get at any of um, the the maize here would have been to break this open with a hard object. Other examples, the curbid family, for example, this includes your melons, your watermelon, your zucchinis. Sorry, I misspoke. I think I included the zucchinis and cucumber with um, the cruciferous family. They belong to the cucurbit family. Sorry if I misspoke. Either way, we have watermelon, for example. And here, looking at our example, I'm just going to highlight something here. The only way that you would get into uh, natural watermelon, okay, approximately 3,000 years BC, or what, 5,000 years plus before the present, would have been through a hard or sharp object. 50 millimeters in size, diameter, extremely bitter, very different from the so-called artificial or the artificially selected watermelon that we have today in 2014. Even in the 1600s, watermelon did not look like it did, and it would have caused inflammation. So 1645, watermelon, watermelon today, and the 1645, this is about the size of a golf ball, even though they look pretty much the same. So some very different food that we had historically. And we need to, as I've pointed out, understand that it did have a different type of impact on our body until we started to select for the certain varieties that we wanted, the certain types of flavors that we wanted. And eventually we started to select out the inflammation, for example, artificially, by reducing the bitter substances in modern watermelon. Okay, so zucchini, this looks pretty small. This would be fairly large in comparison to ancient or pre-domesticated zucchini. Changes that we have in cucumbers over time from the Hartwicki, for example, to a type of English cultivated cucumber. And if you've ever had the opportunity to pick a small cucumber, for example, off of a vine that you're growing in your backyard, sometimes you can really get that bitter flavor out of them. That's what people would have tasted historically. So one of the questions we do have is, why would people eat bitter substances, things that were really repulsive? One of the ideas is, as we know, as we went through the Neolithic Revolution, and just before that Younger Dryas, Mesolithic period, people were going through a lot of nutritional stresses as the environment was changing drastically. Large game was being reduced, and eventually the grains, for example, and the plants that we eventually domesticated, they started to become more available, more abundant in an environment that wasn't as dry and cool as the past during the last glacial major, and with a warmer, wetter environment, it was conducive to growing these crops. All right, so what are we looking at here? Looks like a weed to me. Absolutely not. This is a pre-domesticated form of a food that ultimately is connected to, for example, broccoli, leads to cauliflower. This is cabbage. So some drastic changes have occurred over time, and in the pre-domesticated form, this would have been very bitter. If you've had rapini, for example, and a very bitter variety of rapini, that's the type of flavor that we should imagine we would get from ancient cabbage. All right, what are we looking at here? You're probably thinking, I have no idea. This is celery. Again, some drastic changes have taken place, and if you think sometimes we get a variety of celery that just doesn't taste right, it's bitter, that would be closer to its natural form. Okay, so what's this? These are ancient tomatoes. And again, ancient tomatoes would have been problematic. It's a type of nightshade. It does cause inflammation. Some people have, for example, allergies to histamine. We tend to find more histamines in tomatoes and eggplants. Again, historically, the flavors would have been much stronger as a warning that this isn't something that's entirely good for our bodies. Like I said, there's this idea in biology called use it or lose it. So the current theories that we have here for the TASR2 deletion that ended up taking place was we needed to use it or lose it. A couple of the examples that we see from an evolutionary perspective includes blind fish, for example, that live in areas where there just isn't any sunlight. Their eyes are not needed, so that particular gene was deleted. The same thing is true of carnivores. They didn't need the TAS gene, and they have relics in their genetic code of that particular gene where it was ultimately deleted out. As I've explained, the accumulation of mutations on any particular gene that's not regularly used, this can destroy its ability to make working proteins. So it is in the advantage from an evolutionary perspective, and again, random events, this isn't something that's consciously taking place, that there's a mutation that deletes that particular gene and those species end up being more successful. The other idea here that we have to consider as well is the role that inbreeding plays in gene frequency. So in situations where any particular type of gene, like the TAS2R group, which is inherited in a Mendelian inheritance pattern where this is recessive, so you need to obtain it through mom and dad. You need to be homozygotic for it rather than heterozygotic. Homozygotic, that is only achieved when those two particular genes are inherited in the genotype and expressed in the phenotype. So this would suggest that there were bouts of inbreeding that would increase the frequency of those particular genes, and that does make sense because we know that at about 70,000 years ago, human populations had reduced drastically in size. We were at a near extinction event, and I'll discuss that shortly. We also have a sweet tooth. So alongside of the super taster, non-taster type of analysis, there's a few things that we note. If you're a super taster, you tend not to taste, you tend to say things are too sweet. You tend to think that things are perhaps too salty and too bitter. That does play a role in shaping our diet over time, or our diet, our taste profile, we should say. And individuals who tend to be super tasters do not eat as many good vegetables as individuals who are non-tasters just because of that taste profile. The sweet tooth, well, I'll say something about the super tasters, the non-tasters as well. Non-tasters, they tend to not taste sweet in the same way, so they crave more sweet, they crave more salt. And one of the adverse effects of this non-taster TAS2R deletion is that the likelihood of alcoholism is increased. We have a sweet tooth. So if you're wondering, why do I crave sweet things? Why do humans crave sweet things? They've referred to our cravings for sugar as a type of addiction. Actually, it's a little bit more significant than that. It's actually built into our genetic code to crave sweet things. So this occurred approximately 60 million years ago, 61 million years ago. And again, this coincides with a change that had taken place earlier in our body, where there was not us at this point in time, mammals and the emerging primates that would shortly come along, where there was a deletion in the ability of those mammals to synthesize their own vitamin C. So some mammals, they lose the ability to synthesize vitamin C, and there's another mutation that emerges later on that is just entirely random, and it leads to sugar cravings, and animals seek out sweet things, and that helps them get the vitamin C into their body, and that increases their reproductive success. In ancient times, we also need to understand that excessive sugar would be a toxin, so there was another mutation that emerged in hominins, where we converted sugar to fat. Now, this can also be a problem, as we're aware. On one level, if we use that expensive tissue hypothesis, it does provide us with increased fuel. It allows us to store energy for the future, especially if we're going through bouts of lack of resources. But in modern times, and here I want you to think back to Cordain, over here on the right-hand side, he talked about the evolutionary collision of our ancient genome with the nutritional qualities of recently introduced foods, and he thinks that this may underlie many of the chronic diseases Western civilization faces, and he refers to this as evolutionary discordance. So in other words, while we seem to be hardwired to crave sugar, it's not working out for us today, because we're intaking more sugar, and we're doing so in a way that's leading to increased incidence, for example, of obesity and diabetes, which just didn't exist in the past. Because we also have a lot of changes with respect to the way that we just do things. Most of us are sedentary. We work in our offices. We work at computers. We do a lot of sitting. We do not engage in the same amount of exercise that ancient populations or even individuals in the industrial period would have. So while it was functional among ancient foragers, that fuel, converting sugar to fat, storing that in times where resources were low, today it is quite detrimental. But either way, our sweet tooth, this is a type of adaptation. Takes us a little bit further back, but just slowly working ourselves up to more of the recent changes that we see in the human species. So we're still evolving, right? Yes, we're still evolving. There's no worries about that if you've gotten concerned that somehow we've come to a standstill evolutionarily. Like I said, to understand that evolution continues, we need to understand what's referred to as the Great Bottleneck Event. So historically, population sizes of the Australopithecines, Homo habilis, Homo erectus, they were much larger than we see it around 70,000 years before the present. 300,000 years ago, human populations, they appear to have been quite large. And then 70,000 years ago, when individuals started that movement out of Africa, and that kind of gives us a heads up that something was happening in Africa on a climatic level or resource level. People started to move from Eastern Africa, Southern Africa, and they started to push out to Western Africa. Northern Africa started to make their way over into areas of the around Dubai, for example, in India. We had looked at that last module. China, Australia, eventually began to move into Europe. So around 70,000 years ago, there was a bottleneck event, and this reduced the amount of genetic diversity that existed within the human species. In fact, we got down to the number of about 2,000 to 3,000 individuals, and that's a near extinction event. So this bottleneck, it severely reduced population size, it reduced genetic diversity, and this is why our population geneticists and in molecular anthropology explain the very close similarities that we find, and we've looked at this, we've said all humans, depending upon the test that we're using, the genetic test, we're 99.7 to 99. Sorry, I don't have my Wacom, so this looks a little bit funny, to 99% genetically identical. And that tells us because of those close similarities, we came from a very, very small group of individuals during this bottleneck event, which was about 2,000 to 3,000. Thank you for bearing with me. 2,000 to about 3,000 individuals. And then after that, population size began to increase, and so too would genetic diversity, right? So if we look at changes over time, at around 10,000 years ago, 12,000 years ago, we reached approximately 4 billion, at least the estimates suggest about 4 million individuals. And we have a very slow and gradual climb until we get to about the 1800s, and then we start to increase drastically. I think the suggestion is at about 1800 we had been about 1 billion individuals, and today currently we have hit that, and this is a little bit old data, we've hit that 8 billion individuals in 2022, I believe, 2022, we had hit that number. So in any gene pool where there are more individuals, there are going to be more opportunities for random changes, mutations, single nucleotide polymorphisms, changes in the genetic code that occur regularly. They're going to happen more often in a large population, and there's a likelihood that they will become fixed within populations. So, review. Mutation, single nucleotide polymorphisms, they account for human diversity and diversity in any species. Mutations, they're random events that introduce new features into a gene pool. So, for example, the human gene pool, when we look at the human gene pool, for example, our genotype, 99.9% genetically identical. However, we look at our phenotype, or the outward expression of those differences that exist in our genotype, we see significant differences among populations. For example, there's a lot of variety in skin pigment between and among humans. So at around 70,000 years ago, populations reduced in size, and this helps us explain the lack of genetic diversity in current human populations. Populations eventually began to rebound, and population growth occurred. Microevolution. So think back to the last module, we had talked about microevolution versus macroevolution. Macroevolution refers to the types of changes that we would see, for example, that took place between the Australopithecines that eventually led to Homo habilis, for example, or the development of an entirely, well, that is a new species, a new genus. So that would be a good example, Australopithecines to Homo habilis. In contrast, when we see smaller changes or changes in allele frequency, for example, the distribution of blue-eyed individuals to brown-eyed individuals to green-eyed individuals, and that starts to become fixed in a population. Say at one point of time we have 70% of individuals that have brown eyes, say 20% blue eyes, and 10% green eyes. And then a few generations later, we see that distribution change to, say, 60% brown eyes. We can still say 10% green eyes, but now we have 30%, if my math is right, 60, 30, yeah, 10, say 30% blue eyes. That's microevolution. It's evolution. It has taken place. There are changes in allele frequency. Those changes are becoming fixed within populations because they're serving some sort of adaptive advantage, or they're being selected for sexually. And finally, as I pointed out, more mutations, they're expected to occur in larger populations. So based on this reasoning, human population, we are up to 8 billion individuals. We're going to continue to evolve. We're going to see random mutations. We're going to see new single nucleotide polymorphisms, and they may provide or confer advantages sexually under sexual selection or through natural selection to the populations that they're distributed within. So finally, larger populations, they can eventually be expected to contain more genetic diversity. Now, the other thing that will impact this, we have to consider gene flow. So we're constantly exchanging genes as well, which may lead to eventually further changes in the distribution of alleles, dominant versus recessive alleles. So think back to the Mendelian genetics that we had talked about. Okay, so some recent adaptations. There's a lot of them, and don't try to write all of these down. I'll go through a few of them, and then I'll talk about a few of them in greater depth. But yeah, there's a lot of adaptations that have been taking place over time. So let's go back here, pre 30,000 years before the present. Hair type. Hair type begins to change from the type of texture and how coarse we would have found hair in, for example, African ancestors. So the EDAR hair type. This refers to the changes that we begin to see among humans that are occupying regions of Asia, for example. So darker black, long hair. Longer hair does have a different impact on the ability to retain heat, whereas curly hair tends to allow more thermal regulation for heat to be dissipated. Red hair, we're also going to trace to approximately that pre 30,000 years before the present MC1R gene. And also at that point of time, we have something that's referred to as AMY1. This is a type of an enzyme, an amylase enzyme that aids in digestion that begins to emerge with multiple copies in humans. And we'll say a little bit more about that. So the non-taster, TAS1R gene, N2, and the whole group that appears to emerge around 10,000 years before the present. And a little bit before that, sorry, I've gotten off topic, dry earwax versus sticky earwax, and the body odor mutation. So individuals that tend to have dry earwax, that's more of a recent adaptation, and less body odor. They go together. Early populations and some individuals, they have sticky earwax. That would be a dominant trait, and tend to, when they perspire, there's more body odor. This also performs a sexual selection function. We do emit pheromones when we sweat. So this would have provided earlier a signal, but pheromones attract certain individuals, but currently people think that the lack of odor, that might have also played a role into sexual selection. Blonde hair, and here we're not talking about blonde hair that we might find in Europe, so around the same time in the Solomon Islands, TYRP1 blonde hair emerges. So this is a type of in the model of evolution, we have homologies and we have analogies. As we've discussed, homologies, they come from a shared ancestor. Sometimes species, even within a particular group, they tend to develop the same type of evolutions because they are in an environment that favors that particular mutation. And because mutations are random, it's possible for two of the same types of mutations to emerge at different periods of time in different populations and work their way into the gene pool because they do confer an advantage. Lactase persistence, I'm going to talk more about this. This emerges around 6,000 year mark, 6,000 years before the present. The same thing can be said about blue eyes, just before that 6,000 years before the present. You'll note that blue eyes emerged prior to light skin. We also see mutations in around this period. It's referred to as the FLT1 gene, and this tends to favor women who are pregnant. They tend not to lose the child during pregnancy if there is a malarial infection. So like I said, our bodies are absolutely amazing. Natural selection is amazing. There are a lot of changes that occur over time that do help us. For example, moving up to the top here, malaria resistance. We see that in a variety of different ways in African populations. For example, with two genes here, the DARC and G6PD gene. I know there's a lot of letters here. Don't worry about memorizing these. And also, for example, the hexa gene that we find in populations that tend to have lived in areas that were urban areas. So individuals that have a history with urban areas, they tend to have TB resistance. Same thing can be said about cholera resistance. So at about the same time, we also have the European KITLG gene emerge that's selected for blonde hair among European populations. 3,000 years ago, and I will say more about this, we see some populations with the ability to carry more oxygen in their lungs. And we'll also talk about individuals who tend to have larger spleens and how that impacts them as well. So that would be a change in allele frequency associated with particular populations, conferring an advantage. Okay, our brains are getting smaller. It's true. I'll say more about that as well. CCR5 delta 32. This is a really interesting mutation that occurred at CCR5. This is a chemoreceptor through which HIV enters the body. So individuals, we find, for example, about 18% in Europe have this mutation, CCR5 delta 32, and that provides immunity for HIV. We're not exactly sure when that occurred. Some people speculate in the 14th century. Some people say just a little bit earlier than that. We are getting cooler. We have lighter bones. We're retaining an artery that usually disappears after birth. And recent research, for example, in telomere, little ends on chromosomes, for example, here are the chromosomes, little telomeres at the end. For a long time, they were thought to be non-functional, and now they're being related to intergenerational trauma and life expectancy as well. So we are evolving. Don't worry. Okay, so you don't need to know the Glasssteer formula, but I just want to kind of preface this. When I teach forensic anthropology, a lot of students, when we talk about the Glasssteer formula that works from a normal body temperature of 98.4, we're able to arrive at the how long since the individual has died or been deceased by working from 98.4 as the normal temperature and measuring the internal temperature of an individual, dividing that by 1.5, and that gives us a particular time frame for how long they've been deceased. For example, 2 hours to 10 hours. Students usually say, well, wait a second, 98.4, that's not our average temperature. I'll say absolutely. So when the Glasssteer formula was developed, this was in the 1800s, And we continue to use that number unrevised because that's what Glaster had come up with. But I will tell you that we are getting cooler. So we're cooling down. So the mid 1800s, the average human temperature was recorded at 98.4 degrees across population. So this was the average. However, currently we're seeing something that is coming in around that 97.9 mark, our current averages. Now, this might have something to do with the types of food that we're eating, the changes that we have in agriculture, for example, the reduction of bitter substances, inflammation. So if we're eating historically foods that tended to cause more inflammation or had diseases that caused greater amount of inflammation, temperatures would certainly be higher. So changes in human epidemiology, that could be one of the factors that we're seeing these changes over time. They've resulted in less inflammation. The other thing is inflammation burns calories and increases metabolic temperature. So historically, we can expect to see, with more inflammation, higher metabolic temperature. With the current numbers that we're seeing, and again here think about Cordain, 97.9, this means ultimately that we need probably about 150 less calories per day. Back to Cordain, we're not seeing changes in evolutionary patterns in modern times. So for a variety of reasons, we're probably getting too many calories just because of the changes that have taken place. All right, so a couple of the mutations within the last few thousand years, FADS2 and the TB hexa flaw mutation. So the first one, FADS2, it refers to a type of dietary gene that we find fairly high mutation frequencies in populations in India, specifically with individuals who have a diet that is based upon non-meat sources. So one of the things that we know about meat, it provides us with omega-3 and 6 fatty acids. This mutation allows individuals or helps them to process omega-3 and 6 fatty acids from non-meat sources. And when it's compared to individuals who do not have the same mutation, that goes along with them having a diet that does use meat, so therefore those sources are being obtained through meat, omega-3 and 6 fatty acids. With respect to the hexa gene, this flaw mutation that occurs on at Tay-Sachs, Tay-Sachs is ultimately the result of an enzyme deficiency. So this flaw mutation, it produces an enzyme deficiency, and that results in the inability to break down certain fatty acids that attach themselves to nerve cells and leads to a disease referred to as Tay-Sachs. Tay-Sachs would follow the typical Mendelian inheritance pattern. So this, when you have it in a homozygotic form, you've inherited the flaw mutation on the hexa from mom and dad. An individual will develop Tay-Sachs. We tend to see, as I've said, certain populations in urban centers with higher frequency of this particular gene. Now, the interesting thing, and like I said, so urban populations, the interesting thing is when an individual inherits the hexaflaw mutation only from mom, so it would be a heterozygotic inheritance, that provides protection for TB. And this would also be not with respect to the hexa gene, but this also is the same for cholera and cystic fibrosis. So some really interesting connections have occurred over time, and I'll also point out the same would be true of the sickle cell trait. So if you've inherited it in its homozygotic form, it leads to sickle cell anemia. If an individual has inherited it in its heterozygotic form, there's resistance for malaria. OCA gene. Okay, so the OCA2 gene, this is thought to have emerged as late as 10,000 years before the present and as early as 6,000 years before the present. Typically, individuals associate blue eyes with light skin, and the connection is often made that blue eyes conferred some advantage when it came to UV radiation. So the same type of theory that goes along with that SLC24A5A111T gene that we had talked about. It's explained that this OCA2 gene resulted from a single mutation event. And what the OCA2 gene does is it codes proteins to produce melanin. And therefore, in this particular type of mutation, it shut down the amount of melanin that was produced in eye pigment, leading to brown eyes. Like I said, we tend to make that connection with light skin because we do find populations with lighter skin with higher frequencies of blue eyes than individuals with darker skin and blue eyes. But blue eyes first emerged in individuals that had darker skin pigment. We know that the SLC24A5 gene, that light skin pigment, it doesn't emerge until after blue eyes have. So brown eyes, eventually they're coded as blue. Some people say that there's absolutely no evolutionary, from a natural selection perspective, known. Some people say maybe seasonal affective disorder, so more sunlight into an individual's eyes. Some individuals argue that blue eyes are better at identifying objects that are stationary, so objects' persistence, and that would have aided individuals who were foraging, for example, and locating objects in forested areas. In all likelihood, it probably is a result of sexual selection. That might best explain the persistence of this mutation. It was novel, it was interesting, it was perhaps seen as sexy. Because blue eyes are inherited again through that Mendelian type of pattern where you need to have two recessives, this would suggest inbreeding. Populations that were engaging in endogamous relations. That's the only way that we would be able to see blue eyes expressed at a higher frequency within any population. The gene might persist, but in all likelihood, it would be covered up by the dominant genes at that point of time. So two individuals with blue eyes would go on to be responsible for the increased frequency in that particular allele. EPAS1. I used to climb, and I was always amazed by the fact that Sherpas, for example, in Tibet, were doing an extraordinary amount of work in comparison to the rich Westerners that were going to climb Mount Everest, right? And here we have a Sherpa climbing Mount Everest, carrying all of the gear of the guy who's paid, or the female who is paid to climb this mountain. And one of the things you'll notice is that he is not using an oxygen mask, which a lot of Westerners do because they are just not adapted to climb high altitudes. You know, really at kind of that 6,000 meter mark, we start dying. We develop hypoxia, and that leads to altitude sickness and eventually death. So at around 3,000 years before the present, population geneticists, anthropologists, they have pointed out that there was kind of a broad sweep in the areas of Nepal that led to the ability to carry more oxygen in one's lungs. So Tibetans, for example, individuals that tend to live in higher regions, not the lower regions, they can carry more blood in their oxygen in their blood, and this is induced by hypoxic types of situations. So larger lungs, increased hemoglobin regulation, and this is apparently the fastest genetic sweep that has been seen. I don't know. I'm just reporting the facts here. It's thought that this was inherited from the Denisovans, which would be a parallel group to Neanderthals, and increased selections around 3,000 years before the present. And as I said, it's active in hypoxic types of situations. So where hypoxia is induced, lack of oxygen, the gene becomes more active if present in the individual, allowing the lungs to carry more hemoglobin in the blood. Okay, our brains are shrinking. Yeah. Um, last 30,000 years, apparently about the size of a tennis ball. We think that this is related to the role of society and that culture or knowledge, for example, is being kind of stored in society. So think back, for example, to ancient Sumeria and the invention of cuneiform writing and moving from fairly simple types of documents or recording economic transactions to telling stories. Eventually, the libraries, for example, of Ashurbanipal, they emerged storing mass amounts of information. If we think ancient Egypt, again, hieroglyphics, eventually the hieroglyphics are written on paper, papyrus, and knowledge is stored. There's no need to carry as much information in our brains as we did historically. So the research suggests that this appears to be connected to advances in writing and changes over time, and our brains are shrinking. I can only imagine what's happening with in modern times with digital storage of information and how much information we're expected to hold in our brains in comparison to what we needed to hold in our brains 10 years ago or even 5 years ago. There's significant changes taking place. Okay, so this is where I left off in class, the milk revolution, and I will pick up in our next segment after it's taught next week. Take care.